Hello and welcome to my podcast. My name is Nika. I am a future doctor and scientist from Georgia, and I'm so excited about today's episode. I have the privilege of hosting Professor Thierry Berney, a world-renowned surgeon whose work has shaped the field of transplantation. He has received multiple international awards, published over 350 scientific papers and book chapters, and today, he serves as a professor at the University of Geneva. Professor Berney, thank you very much for joining me today. To begin with, could you share what inspired you to specialize in transplantation science and surgery?
Okay, so first of all, thank you, Nika, for having me, and hello to your audience. So, what inspired me to join the field of transplant surgery? When I finished my medical studies, I always thought that it would be important before going into the clinical field to have a scientific background. And I did 2 years of basic research in the field of immunology. And I also always knew that I wanted to become a surgeon, and the obvious link between immunology and surgery was to go into the field of transplantation. So that's what I did. That's how it all started, and then I had a regular residency training, and very rapidly decided to go the way of transplantation.
Professor Berney, for our listeners, could you explain what pancreatic islets are and also why are they so important for blood sugar control?
Okay, so you are going straight to 10 years forward since the end of my studies, because of course one of the fields that I decided to specialize in was islet cell transplantation. This happened because during my at the end of my residency training, I went to get some specific training in organ transplantation in Miami, in the US, where I spent 4 years. I spent 2 years doing some research program, scientific research, and 2 years being trained as a transplant surgeon. During my research, I was very lucky to be part of one of the most active centers in islet cell transplantation, and that's what really led me to keep this field as my main field of not only basic science research, but also clinical research and clinical activity. So, what are these islet of Langerhans? Islet of Langerhans they are called islets because they look like little islands of cells that are scattered throughout the pancreas. And in these islets, there are cells called the beta cells. And the beta cells are very important because they secrete insulin. And when these beta cells are destroyed by some mechanism that is abnormal, obviously, the islets are not able to produce insulin anymore. And this causes a disease that is well known to everybody that is called diabetes. More specifically, type 1 diabetes or juvenile diabetes, because it occurs more often in youth and adulthood. So this is what the islet of Langerhans are, and this is why they are important.
Uh, Professor Berney, when we talk about uh, islets, we, I couldn't leave this subject without asking this question. Why are islets, and especially beta cells, destroyed in type 1 diabetes? And why is restoring insulin production so difficult?
So the islets are destroyed in type 1 diabetes because the immune system, immunity, uh, gets activated in the wrong way. It's a dysregulation of the immune system. And it's the immune system that identifies these beta cells as foreign agents, if you wish, and they are attacking these cells and destroying them. And this takes a few years to develop, but at the end of the process, there is absolutely no more beta cell available in the pancreas, and therefore these patients, they produce no more insulin, and they have high blood sugar levels. And the only way to go against that, I mean, immediately, is to give injections of insulin. So there are lots of different types of insulin that can be given. They can be given with injection. They can be given with the help of automatic pumps, but you still need to have injection of insulin. And this allows to have an okay blood sugar control, but it's not perfect. And in some patients, it works very, very well, but in a few patients, it does not work that well because the blood sugar level remains very difficult to control. And these patients are patients that ultimately will be a candidate for something more than replacing insulin. And this is replacing the missing cells. And you can do the replacement of the missing cells either by transplanting a whole pancreas or by isolating islets from a pancreas and transplanting the islets in these patients. So transplanting the islets seems more logical because you only transplant what is missing, what is needed, but it's technologically advanced. It's quite technically, I mean, difficult to do. You need some real know-how that is developed over years. And also, it does not work quite as long as a whole pancreas transplantation for reasons that are not always very well understood.
Professor Berney, uh, how does an islet transplantation actually work in practice? And also, what are the main obstacles? Graft survival, immune rejection, or there should be something else?
Yeah. So an islet transplant, as I told you, you have to first have a donor. So it's the same as a kidney donor, a liver donor, a pancreas donor, a heart donor. It's a deceased donor. It's a donor that is usually in a situation of what we call brain death. So when we have such a donor, we can remove the pancreas, and depending on the donor, but also depending on the recipients that we have on the waiting list, we decide to either perform a whole pancreas transplant or to isolate the islets from the pancreas. This is done in a specialized lab. And these islets are isolated, purified, prepared, and they are conditioned in a bag that looks very much like a blood transfusion bag. The interesting thing is that these islets, when you transplant them to the recipient, we transplant them into the liver. And why the liver and not the pancreas? It's for many reasons, but the main reason is that the liver is easy to access, and the islets are injected into a venous blood flow that has a low pressure, so they are not harmed by a hard how can I say by a burst of high blood pressure, like if you injected them in an artery. So that's the way we do it. Now, these islets, they are not injected in their normal environment, and of course, this causes problems. So, of course, these islets can be rejected, and this is the case of any other organ. So it's not any different to a kidney or a whole pancreas. To prevent rejection, what you have to do is to give immunosuppression, anti-rejection treatment, and these patients have to take that for the rest of their lives. And again, this is exactly the same for a kidney transplant, for example. The second thing is that they are in an environment that is not their natural environment, and they probably have to withstand some inflammatory phenomena that, as time goes by, slowly exhausts them. So this is the reason why islet grafts do not function quite as long as a whole pancreas graft, for example. So there's lots of work that is being developed in order to mitigate these problems, and lots of progress have been made over the past years.
That's really interesting. And Professor Berney, when you first started your career, what did the field of transplantation and islet transplantation look like compared to now?
Okay, that's a very good question. And there have been a lot of progress because, as I told you, when I started to work really in islet transplantation, we had an islet transplant program that was starting at the University of Geneva, but where it was really the start of the activity. And at this time in Geneva and everywhere else, the results were not very good. We could have about 10% of our patients without insulin at 1 year after transplants, which was not really good. When I was in Miami, at this time, there was a very important thing that happened in the field. It was what is called the Edmonton Protocol. Edmonton is a city in Canada, in northern Canada. And in Edmonton, they developed a protocol where they found out that in order to improve the results, you should probably inject a higher quantity of islets, and you should adjust the immunosuppression, the drugs that you give, because some of these drugs are a little bit toxic to the islets. And with that results, all of a sudden, they went from 10% of insulin at 1 year to more than 80% of insulin. So this was a major game changer in the field. And this happened when I was in Miami, like right in the middle of the high activity centers. And this is sometimes that we started to apply when I moved back to Geneva. We're talking about the end of the 1990s, beginning of the year of the 2000s. When I got back to Geneva, we had also adopted this protocol, adjusted it, and we managed to obtain similar results. And over the past 20 years, the results of islet transplantation have constantly improved. Also because of technical reasons, because our islet isolation manufacturing process in the isolation lab has also improved. The reagent that we use have improved. So many things have improved. And now we are really reaching really good results with always like 80% of insulin at 1 year, but up to 40, 50% of patients still without insulin after 5 years, which is quite remarkable compared to what we have in the beginning.
Professor Berney, you mentioned immunosuppression, and do you think tolerance-inducing therapies or stem cell approaches could replace these lifelong immunosuppression?
So this is this is a vast question, and we could be speaking about that for a whole day. But so immune tolerance is what is called what we call the holy grail of transplantation, because if we are really able to educate the immune system so that they stop recognizing a transplant as foreign, then we would be able to have organ transplanted without lifelong immunosuppression. This has proven a very elusive task to accomplish, because we still need immune suppression in these patients. Now, can stem cells be a response to that? Maybe. There are groups who are working on genetically modifying the stem cells so that they become hypoimmune. So there are some trials that are ongoing right now, and that could be promising, but they are really, really at a very, very starting stage. So it's difficult to say if this is something that will be a possibility in the future. Now, talking about stem cells for islet transplantation, one of the problems is, of course, the problem of organ donor shortage. And if you could generate islets from stem cells, you could theoretically have an infinite source of pseudo islets or tissue producing insulin. And there is currently a trial that is ongoing in the US and in Europe where with a product that has been generated from embryonic stem cells that has been differentiated into endocrine cells, so insulin producing cells, but not only insulin, also the other cells of the islets that produce other hormones. And these cells reaggregate and they make structures that really very much look like regular islets. And this trial, which is ongoing at the moment, has had extremely good results with like, I don't remember the exact number, but it's more than 80% of patients are off insulin after 1 year with excellent blood sugar control. So this is something that has to be followed, and we will have to see what will be the next stages of this, because the big question is first, we have short-term results and they are absolutely spectacular, but what will be the long-term results? That's the first question. The second question is, how much will it cost, and will it be affordable for a healthcare system to administer these cells in in a large scale? And finally, also talking about large scale, how scalable is it? Because so far there have been something like 20 patients transplanted. Their trials aims to go up to 50 patients, but there are much more patients than that waiting for something to happen and living with type 1 diabetes. So these are the questions that have to be answered. But nonetheless, there are spectacular progresses that have been made with stem cells.
And Professor Berney, I remember during the pandemic, uh, people with diabetes were suffering because they were in a very harsh condition. Uh, some of them had transplanted pancreas, and they were on immunosuppression therapy, and some of them couldn't get the vaccine, and who did? I think the vaccine was less effective, as far as I remember.
Yeah, so this is true. That there have been lots of studies that have been published about that, and about the immunosuppressive drugs. There was in particular one that was pinpointed as being responsible for a lower efficiency of the vaccines. But by and large, there have been a lot of literature on that. Patients, I mean, transplanted patients, and not only of islets, of any organ that were transplanted, that were vaccinated against COVID, they had much better outcomes because they had a much lower risk of catching COVID. And if they caught it, they were developing some antibodies still, not always at the level that we would have liked, but there was some protective effect. That's clear.
Sure, I think that's really important for this kind of harsh situations. And alongside with tolerance-inducing therapies, uh, also stem cell-derived beta cells are a hot topic. And do you think they can fully replace donor islets in the future, at least?
I don't know. I mean, we're back to what I said before. It's a matter of scalability. Is it possible to produce them in sufficient amounts so that they will be available to all patients with type 1 diabetes, because that's the ultimate goal, to be able to transplant patients before they start developing complications. But if you want to transplant everybody, you have to have a risk-benefit ratio that is favorable. If you transplant everybody, but they need lifelong immunosuppression, it's not going to be a favorable risk-benefit ratio for everyone, but only for those who have complicated diabetes, because immunosuppression comes with some side effects and some risks. So what we need is to really develop at the same time scalability, development of hypoimmunogenicity. That means no need for lifelong immunosuppression. And once this is done, what will be the cost? Will it be affordable for a healthcare system? At the moment, these stem cells are produced by private companies in the US. And of course, companies, they have made investments, and they need to have a return on investment. And for this reason, we can anticipate that the cost might be a little elevated. The more they are able to produce, probably the less the cost per graft will be lower, but the overall cost will still be quite important. So we need to. This is something that is going to be important: cost control.
And Professor Berney, you mentioned about genetically modified organs and also uh, bioengineered. And do you think these bioengineered or even artificial organs solve the shortage of donor organs? And also, how might the modern CRISPR and gene editing help create universal donor islets of organs? I mean, universal.
Yeah, no, that's a very good question. So artificial organs, meaning organs that are made totally with artificial materials. So if you think about it, a dialysis system for kidney transplant is an artificial kidney. So there are people who are working on a model of a miniaturized and implantable artificial kidneys. So I don't know when this will be feasible, we will reach, but this is something that that clearly has potential. Uh, if there are some nephrologists looking, they will kill me, but the kidney is a relatively simple organ compared to the liver, for example. The liver is a whole laboratory, and reproducing all its function with an artificial organ is going to be extremely complicated. Implantable heart, these are things that exist, so and people are working on it. So this is another thing that is probably possible. So talking about artificial organs, they already exist in a way. They need to be miniaturized and made easier to implant and easier to tolerate. Now, talking about bioengineered, and you're talking about genetic modification with the CRISPR-Cas system. So for your listeners, the CRISPR-Cas system is a system that allows to very easily remove some genes from an organism or add some genes to an organism. And you can multiply it quite indefinitely. This is something that is used and has been used for xenotransplantation. I think everybody has followed the activity of xenotransplantation of hearts or of kidneys in the US and elsewhere. So xenotransplantation means transplantation of organs obtained from another species. In this case, it's pigs. And these pigs have been modified with the CRISPR-Cas system, And these pigs have 10 genetic modification, all done using the CRISPR-Cas system to make the organs easier to tolerate and by the recipient. So the results have been a little bit mitigated, and there is still some progress to be done. But this is something that is already applied in the field of xenotransplantation. Now for islets, this is also something that has been applied. There have been very recently a paper published of a first case where they transplanted islets that were modified by a CRISPR-Cas system to remove the antigenic determinants. And these islets, they have been able to demonstrate that these islets were not rejected after several months of implantation because they were not recognized by the immune system. So again, this is very, very first stages, and it's going to take a long time before it is upscalable. But these are experiments, if you wish, done in human beings that are extremely
Sensitive.
yeah, promising, and I think we will see a lot of progress done in the coming 10 years.
And Professor Berney, kidney transplantation is the most common type of transplant worldwide. What do you see as the biggest scientific and medical challenges that remain today?
Okay, so scientific. I don't think there are that many scientific challenges for kidney transplantation because we have reached now results of of kidney transplant survival of more than 90% of this kidney, 95% of kidneys survive after 1 year. What is the biggest challenge is more the access to transplantation because there are so many people on dialysis who need a kidney transplant and who don't get it. And so this is, I would say, the biggest challenge. How to overcome this, I think, and Georgia is a country that has a big problem in that regard because to do organ transplantation, you need to have organ donors. And there is in this country a law about organ donation and transplantation, but it's not applied very well because there is no real government agency from the Ministry of Health that is in charge of putting together a national waiting list, putting together some allocation rule, putting together national coordination of the donors. And this is something that really should happen. There is a law on transplantation that is very well done. There is one person in Georgia, Dr. Gia Tomadze, who was trained in the US in the late 80s and early 1990s. And since he has been back in Georgia, has worked relentlessly in improving the situation, in obtaining a law. And Georgia has a very modern law on organ transplantation that was voted last year by the parliament. But unfortunately, it is not really implemented, and there is no availability of deceased donors. So in Georgia, what you are relying on is live donors from friends, from family, etc. And unfortunately, people are not moving forward enough to donate their kidney to their siblings, to their parents, to their brothers, sisters, families. And I think this is something that should be done. And probably one of the major reasons for that is that the people on dialysis, they are reluctant to ask their family to donate. And the donors, the potential donors, they are reluctant to donate because they think that it's going to hurt them to donate a kidney. And this is something that really we have to work a lot about, give some information to the patients and the families that if you do a proper donor selection, you can donating your kidney to one of your loved ones is a very, very safe procedure, and is not associated with problems, with health problems in the even in the long term.
I'm sure these issues regarding the transplantation and the lack of support of the population is very touching for people seeking for help. And Professor Berney, are there lessons from islet and pancreas transplantation that could be applied to kidney transplantation, for instance, or is it vice versa?
But I think that what they have in common, islet, pancreas, kidney, is that we have waiting lists and we have more persons in need of organs than persons who are able to receive one. And the lessons that we have learned from one another is, in immunosuppression management, and but the issues are pretty similar, I would say. Of course, each organ is specific, but I don't think there is that much. It's the whole field of organ transplantation that is important. That's why I think it's important to have some transplantation centers that perform all kinds of organ transplantation, because the more organ you transplant, the more exposure you have to the potential complications of immunosuppression, for example. When I was in Geneva, I was the chief of the Department of Transplantation in Geneva University Hospital, and in this hospital, we are transplanting islets, pancreas, kidney, livers, and what is important is that all these patients, they have issues of complication that are similar. And the more you see, the more experience you get, and the better you can treat them. So I guess that's the way there are lessons to be learned, is about mostly the infectious complications of immunosuppression, for example, how to manage them, because many of them are preventable and many of them are treatable.
And Professor Berney, in this technological era, do you see AI and machine learning playing a role in transplantation medicine and surgery?
Oh, absolutely. It's already playing a big role. Artificial intelligence, there are models that have been developed for many things, for uh, donor assessment, organ allocation algorithms, uh, prognosis of kidney grafts, for example. It's mostly been developed for the kidney, but it's also something that is being developed for other organs. So what we are talking about are algorithms developed thanks to artificial intelligence in transplant medicine. Talking about transplantation surgery, the most obvious example of artificial intelligence in surgery is the use of robotic surgery in performing transplants, or in performing organ procurement. So for live donors, uh, robotic surgery is a standard for in many hospitals for the procurement of live donor kidneys. There are many centers that do live donor kidney transplantation with robot assistance, especially in obese patients in which there are many challenges in the surgical approach. And there are also centers that are doing for live donor liver transplantation, that are doing procurement of the liver with robotic surgery, and even transplant of the surgery by robotic surgery. So this is something that is already used and that is going to develop significantly. So yeah, absolutely, that's a very good question.
And also, Professor Berney, uh, As far as I know, the shortage of donor organs remains one of the biggest barriers. What realistic solutions do you see as a surgeon in the future?
What are the what?
What realistic solutions do you
A
see
solution.
in the
But
future?
the thing is, you,
I
what
mean, the shortage of donor.
yeah, I understand. To do organ transplantation, you need donors. You need deceased donors, brain dead donors, or donors with cardiac arrest. So thank goodness, intensive care units are getting better and better at reanimating these patients and at preventing them of dying. So the impact that it has is that there are less patients that are available for organ transplantation. I'm talking about deceased donors. And of course, this is a blessing because the first thing that we should do is care for the patients. But in a country like Georgia, what you need to do is really develop structures that allow for deceased organ donation. And then the second thing is to uh, to develop the live donation of organs. And this is something that is feasible for kidney, for liver. It has already been done for the pancreas in Asian countries, in Japan, Korea, but also in the US. So these are things that can be developed. Obviously, it cannot be done for the heart, but this is something that can be done. Develop, making some information to the population. This is what has to be done. And one of the major things to be done is prevention. If you can prevent to have all these patients in the waiting list for an organ, you have to try to prevent the causes. And if we want to speak about kidney transplantation, about more than half of the patients on dialysis in Georgia, but it's not only in Georgia, it's the same in the US, for example. They are they have kidney failure because of diabetes or I'm talking about type 2 diabetes now, not type 1. Type 2 diabetes, high blood pressure, or both together. And this is in almost all cases associated with overweight or obesity. There is a rate of obesity that is quite high in Georgia, and this is something that has to be worked on. Preventing obesity, promoting lifestyle changes, and promoting early if you have diabetes and high blood pressure, being treated efficiently. Many patients are not really treated very efficiently because they don't go to see their doctor early enough. But the most important would be to promote to promote, sorry, lifestyle changes to prevent
Healthy lifestyle habits.
obesity, overweight. This is very important.
And Professor Berney, you have worked extensively in Switzerland and now in Georgia. What differences have you noticed in the way medicine and research are approached in these two countries?
So I think the main difference between two countries is a question of wealth. Switzerland is a very wealthy country, is one of the top five. I think, I'm not sure, and has a lot of resources, financial resources, to develop research programs and also for healthcare coverage. Um, Georgia is oscillating between high middle income and low middle income. I think that at the moment it's more in the low middle income situation. So, finding resources for research promotion or healthcare coverage is not as easy, but it's easy nowhere, but it's more difficult in Georgia than in Switzerland. I think that the healthcare system should be developed a little bit better. I think there is potential in Georgia to have better healthcare coverage. The way it works is suboptimal, and I think the Ministry of Health should really work in that to be able to allow mostly better reimbursement of patients and procedures, because one of the reasons why patients are not always well treated is because a lot of you know diagnostic tests or medication are not reimbursed, and patients cannot afford it. So this is something that has to be worked on. I was talking about the important rate of obesity. And there are really, so we are talking about prevention, but when you have patients who are obese and want to lose weight, diet is not always very efficient in everybody, because you need to have a mindset for that. But there are some easy ways, which is bariatric surgery, or now the new drugs like you have heard about Ozempic or Mounjaro, etc. Neither of these is covered by the healthcare system in Georgia, and this is something that it should be given some thought, because it's a major healthcare issue. If you manage to make this patient lose weight, they will be healthier, they will work better, they will work more, they will the whole society will benefit. At their family level, they will benefit, but at the country level, it will be beneficial. So I would advocate for that. These are drugs that or procedures that are reimbursed in Western countries, not only Switzerland. Of course, not anybody, but you have to have an indication. It has to be indicated by a doctor, and there are some criteria for that. But when you meet these criteria, you should have the possibility to benefit from bariatric surgery or these drugs like Ozempic or from the same family.
Professor Berney, thank you so much for sharing your knowledge and vision with us today. We have covered the science of islets, the challenges of transplantation, and the exciting future possibilities, from stem cells and the gene editing to bioengineering and AI. Thank you again. And to our listeners, I hope you enjoyed this episode. Stay curious and see you next time.
Thank you very much, Nika, for having me, and thank you to everybody for listening to us.
Thank you.